morning, everyone. Today I will present the European Multi-Center Study CZ2, which we are conducting with 7 European hospitals. So as you all know, in epilepsy, accurate seizure detection is very important in order to optimize clinical management. The current gold standard to do that is the video EEG monitoring. The patient has to come for this to the hospital. It requires bulky equipment. It's quite costly, and it's also only a snapshot in time. So very long-term monitoring is not really feasible. Now, when the patient is at home, so for the majority of the time, the neurologist has to rely on the seizure diary. And as you all know, the patient-reported seizure frequency is quite problematic, as underreporting is a very common problem. So to overcome this and to have more objective seizure monitoring, also at home, we are evaluating the Sensor Dot, which is a multimodal wearable developed by Biteflies, which is a Belgian company. In the first phase, we are evaluating the Sensor Dot in the hospital in patients with absence seizures, focal impaired awareness seizures, and tonic-clonic seizures. In the hospital, we have the ground truth, which is the video EEG, so we can compare the seizures to that. Now, in the second phase, we will evaluate these 3 seizure types, or the Sensor Dot in these 3 seizure types at home. There we don't have the ground truth, and we have to rely on the seizure diary. Currently, we have focused on the absence seizures, as you don't need a lot of patients to have like a nice amount of absence seizures. So this is the Sensor Dot, which is developed by Biteflies. The recording device is this small thing. So this is put into the cradle and then attached via an adhesive to the back of the patient. And then we connect the electrodes behind the ears to the Sensor Dot. Now, depending on the type of epilepsy, we have different configurations. So the setup you see here is a generalized setup. And here we have 2 electrodes on both sides of the head. This creates then 2 ipsilateral bipolar channels, so one left and one right. Now, if we have a focal setup, then, for example, if we have a focal left, then we'll have 2 electrodes on the left side and one on the right side, so that we will have 1 ipsilateral left channel and 1 cross-head channel. The Sensor Dot is also a multimodal wearable, so we can also measure EMG and ECG. And that is with the second Sensor Dot, which is placed on the chest and connected to the electrodes on the deltoid muscle for EMG and to the electrodes on the chest for ECG. The Sensor Dot also inherently has an accelerometer and a gyroscope to measure movement. And here is an example of how you can see it on the patient. So we have currently focused on the typical absence seizures. And for that, the patients came to the hospital for routine video EEG monitoring. And at that time, we also asked them if we could measure them with the 2-channel Sensor Dot. For the typical absence seizures, we only use the EEG dot. And we also asked them to keep track of the seizures in their seizure diary. The 25-channel video EEG was annotated by the treating neurologist in the hospital, while the 2-channel Sensor Dot EEG was blindly reviewed by 5 central readers, which were all epileptologists. We then compared the seizure annotations on the Sensor Dot EEG to the annotations on the video EEG, as well as for the seizure annotations on the seizure diary to the video EEG. And we defined the true positives, the false positives, and the false negatives. And from this, we calculated sensitivity, precision, and F1 score. And the F1 score is a general measure which takes both sensitivity and precision into account. And this enables a better comparison between the different seizure detection devices. Here you can see an example of an absence seizure as detected on the Sensor Dot EEG in the hospital. So the upper panel is an absence in a pediatric patient. You can nicely see the 3 Hz spike and wave discharges. And the lower panel is an absence seizure in an adult patient. So blind review of the full 24-hour Sensor Dot EEG resulted in a median sensitivity of 81%, a precision of 89%, and an F1 score of 73%. But most importantly, the review time was 1 to 2 hours for every epileptologist, which is, of course, much too long for it to be adopted in clinical practice. That is why we collaborate with engineers from the University of Leuven And they have developed a semi-automatic absence detection algorithm, which goes through the entire file and flags segments of interest, so regions of which it thinks it is an absence seizure. And then in the second time, we gave those files with the segments only to the central readers, and they read those files, and this led to a median sensitivity of 83%, precision of 89%, and an F1 score of 87%. And this is mostly the case because the scores became much higher and the ranges are more narrow. So reading those segments, you can have a much more focused review. And it's also a bit of a learning effect because they now know what the seizures look like on the sensor.eeg. So this led to a reduction in review time to 5 to 10 minutes. And the whole concept of the sensor dot is that the patient attaches the sensor dot, they measure for 1 day, then places the sensor dot back into the docking station where the battery will recharge, and also the data will be uploaded to the cloud. A dedicated algorithm will then go through the files, flag the regions of interest, and these segments are sent back to the treating neurologist, who then just has to decide whether it's a false positive or an actual seizure. So this concept allows us also to use it at home, which we are currently studying for the absence seizures as well. The benefits of the sensor dot are that there is now more objective documentation of absence seizures, which allows us to better monitor the response to treatment. So if we change medication, then we can measure at baseline and after the...
Could you go back one slide to the conclusion?
So we can also measure after we have given the medication and see if there is a difference in seizure frequency. Could you advance one slide, please? And also the sensor dot is a more objective endpoint for anti-epileptic drug development trials. Look into the detection of tonic-clonic seizures, in which the real benefit, of course, lies into the online detection of the seizures, after which it can give an alarm if a patient has a tonic-clonic seizure. And we are also studying the focal impaired awareness seizures, which is a bit more challenging because of achieving high sensitivity together with a low false alarm rate. Thank you.